video, we're going to be looking at several applications of biotechnology. We covered this in 4 lectures in class. They were all pretty short lectures, so I've decided to combine all of this into one giant biotechnology extravaganza. And so first we covered viruses, and we talked about how viruses are very small. I think that's important to understand about viruses. They're non-living. They're just proteins and DNA. Or RNA. Some viruses are RNA viruses, and you see here viruses have lots of different shapes, but essentially they're all the same sort of idea. There are protein coding, and then there's this genetic material on the inside. Some viruses can actually develop this like an envelope around them, and it's not that they develop it necessarily, but they have gathered it from their previous hosts. Their hosts are always some sort of biological organism, some sort of cell, and this envelope can be part of the former organism's cell membrane. And so there's a picture of how that works. Bacteriophages, we'll just mention quickly. We talked about them when we talked about the Hershey-Chase experiment. They're just viruses that attack bacteria, and so you can see here the protein capsid, the DNA on the inside. So how do viruses reproduce? Well, a virus comes in contact with a host cell. The host cell invites it in, and the DNA of the virus comes inside. And that DNA of the virus, once it enters into the host, is called a prophage. And that DNA begins to replicate. Well, that DNA codes for the pieces of the virus, like the actual protein capsid. And so once that virus gets on the inside of the cell itself begins manufacturing more and more viruses. Well, this eventually becomes detrimental to the cell, and as those viruses form, that cell will actually lyse, or actually kind of explode, releasing those newly formed viruses into the environment. And then each one of those will follow in the same suit. And so you can kind of get this idea. If one cell makes 8 viruses, which I'm sure it's much more than that, then each one of those 8 will go and infect 8 more cells, which will make 8 more. And so eventually this gets very out of control. When the virus is in this stage, when it's actively reproducing, this is called the lytic cycle. When the virus is actively reproducing, when it is active, that is the key word. The virus is actively being made and constructed by the host cell and is causing more and more host cells to do that, causing damage to the organism. The virus will do that until the organism dies, or until the organism's immune system fights off the virus. But that DNA remains intact in the host. If that DNA remains intact and goes into a dormant state where it's no longer active, this is called the lysogenic cycle of the virus. Notice on this picture that you have on the right hand side, where in the lysogenic cycle, that prophage, as you can see here, is being reproduced normally by the host cell, just as if it were another part of its DNA, and making more and more cells that have the copy of that virus. Some sort of stimulus can occur, like in the case of humans, when we have some people have a virus that causes cold sores, or maybe the chickenpox virus. I had chickenpox when I was a kid. That virus is in my cells. Well, stress can cause that virus to rear its head again, and then at that point it's called shingles. That virus enters back into the lytic cycle. And so the life cycle of a virus can oftentimes be characterized by the switching between the lysogenic and the lytic cycles. This type of viral reproduction can actually be used to benefit people. If you manufacture a virus, for instance, that has a correct gene on it, take a broken gene, maybe a cancer-causing gene, well, that a virus can be manufactured that inserts the correct gene into those cells. This is called gene therapy. Some viruses actually contain portions of RNA And they have an enzyme in them called reverse transcriptase, which turns that RNA into DNA, and then that DNA infects the cell. These viruses are able to reproduce, are able to mutate much faster than normal viruses. They're called retroviruses. AIDS, or the HIV virus, is a great example of the retrovirus. Also, just simple common cold. So, moving on to part two, getting a little bit more into bacteria. One important thing when we talk about bacteria, here's the size of bacteria again, really small compared to your average cell. Bacteria or prokaryotes have a single circular chromosome, and they have the cell wall and all that stuff that we know. But we know the single circular chromosome. One thing we haven't talked about is bacteria also contains small portions of DNA called plasmids. A plasmid is just another portion of DNA that gives that bacteria some genetic variation. Now, we've talked about ways that bacteria can have variation, but we didn't talk about it in terms of plasmids. Take transformation, for instance, where bacteria take up DNA from an outside source. What is that DNA that they're taking up? 9 times out of 10, it's some sort of plasmid. Transduction. We haven't talked about that, but we have now talked about viruses, so you can get the idea. Transduction is where viral DNA has affected that bacteria. And so we've talked about now the lytic and the lysogenic cycles, and so you can get more of an idea of that. And with bacterial conjugation, I have the word up here. With bacterial conjugation, you have this sex pilus that is connecting one bacteria to another, and they're exchanging DNA. Well, what are they exchanging? They're exchanging plasmids. A new organism is not being created here, but it's just simply exchanging DNA. So that's the way that bacteria can create some variation. Here's another picture of this showing how this plasmid is being shared. Another way that bacteria oftentimes have some variation are something called transposons. This lady, Barbara McClintock, in the 1940s discovered this mechanism for that. It's a way that gene expression can be controlled in prokaryotes. Also now it has been discovered in eukaryotes as well. And a transposon is something that she called a jumping gene. And a jumping gene literally is what it sounds like. It jumps around. How does it do that? Well, there's an enzyme called transposase, as you can see here, that causes that gene to leave its section of the DNA that it's in and enter into another section. And so if that transposase is in a gene, for instance, that gene is no longer being expressed. And so you can have a gene jump around. That's just a basic transposon. There are composite transposons that basically take an entire gene, not just itself, but take an entire gene with it. And in this case, it shows them carrying an antibiotic resistance gene, which could be helpful if you're bacteria. Now, bacteria have a control mechanism for gene expression, and it's called an operon. And an operon is a very simple mechanism in which there are a couple of pieces present. Things that we have talked about are promoter sequences. We understand promoter and RNA polymerase and initiation complex and all that kind of thing. The main portions of the operon that I want you to focus on, you see that in this one best. The repressor is this red protein, and the repressor does exactly what you think it does. It represses the transcription. That repressor will actually sit down on top of the promoter sequence and prevent the initiation complex from attaching to the promoter, thus preventing transcription. When an inducer comes in play, this inducer is this green ball, it causes the repressor to lift off the promoter and then allow transcription to occur. In this particular operon, this is called the lac operon because it codes for a gene that breaks down lactose. And so when lactose is present in the cell, that cell needs to break it down. And so in this case, lactose becomes the inducer. Lactose binds the repressor, causes that repressor to lift off. RNA polymerase binds the promoter, creates the gene or creates the protein that breaks down lactose. Lactose is broken down until the last piece of lactose, the inducer, is broken down, thus causing the repressor to bind back to the promoter, thus causing that gene to no longer be expressed. A couple other things to mention. We kind of went through this really quickly, and that's fine. One, for part 3, we talked about the bacterial cloning process. There's a technology called recombinant DNA. It's a big fancy word for basically meaning recombining DNA. This is where we take a gene that we want to put into another organism. Most of the time that's a prokaryote, but more and more we are figuring out how to do this with eukaryotic organisms as well. And we cut that gene into place. We take a gene, we take a plasmid, we cut the plasmid, insert the new gene, put that into a bacteria, and there you go. How do we cut that gene? There are enzymes called restriction enzymes, and restriction enzymes do one thing: they cut DNA. They cut DNA at a very particular segment. You see here in this picture, GAATTC. Restriction enzyme sees that, it cuts it, creates these things called sticky ends. They're just called sticky because they are attracted to each other. Well, what if we could create a gene that had those same sticky ends and insert that gene into where that plasmid was? Now we could insert that plasmid into a bacteria, and that bacteria would now produce that protein. This is a very common kind of thing. How do we know if the bacteria have the plasmid that we just gave them? Well, the way that they do this is something called a genetic marker. And that genetic marker is usually, you can see here, antibiotic resistance. So you'd have a plasmid that you know contains antibiotic resistance. You cut that plasmid, you insert the gene of interest, this human gene that we want to put in over here. This is black gene. And now this plasmid not only contains antibiotic resistance, but it also contains the gene that we want to teach bacteria how to make the protein of. And we introduce that plasmid into a bunch of bacteria. Some of the bacteria are going to take up the plasmid, others of the bacteria are not. How do we know which ones didn't? We apply antibiotics to the bacteria. The ones that didn't live, or the ones that didn't die, the ones that did take that plasmid up are now resistant to that antibiotic and do not die. And so thus we have antibiotic resistant bacteria, but they're also able to create the protein that we wanted to make. And so that is a very common type of practice that is used in cloning bacteria. Another thing that can occur, let's see, is something called yeast artificial chromosomes. The problem with bacteria is they're prokaryotic and we are eukaryotic, and so one way to create genes, or a lot of genes, and have them replicated is to make a chromosome, to literally make an artificial chromosome where we have a lot of genes together. And the good thing about that is a yeast cell, because it's eukaryotic, will recognize things like introns and exons, for instance, like the 5' cap and the poly A tail and all those things that prokaryotic cells don't do. And so we can create a chromosome and then insert that into yeast, and those yeast will then replicate and create more copies of that chromosome. And lastly, for part 4, just a couple of things to talk about. This guy invented a technique called polymerase chain reaction. Look at the words: polymerase. Polymerase creates DNA and DNA replication. Chain reaction is something that happens more than once, and each time it happens, it builds and gets bigger. Well, basically what he did is he created a system where you can take a single gene, a gene that you want to replicate, and you put it in this polymerase chain reaction. Essentially what you do is you heat the DNA up to a certain point. That DNA separates because of the heat, and then polymerase binds to that DNA, makes a copy of it. Now you have 2 copies. But you do the same thing again. Now you have 4. And you get the idea. This can get out of hand really quickly. Get tons and tons of copies of DNA. This is used a lot today in any sort of research that involves particular genes. So, very, very monumental discovery for the field of biology. Another technique that is used quite often is called gel electrophoresis. And gel electrophoresis is where enzymes, restriction enzymes, are applied to sections of DNA. And those restriction enzymes cut the DNA into segments, and then that DNA is put in a gel like this. So you take the DNA that you've cut up and you put it in here. This end of the gel is very positive, so the DNA is going to want to go through the gel. Well, the really small fragments, because they're small, are going to be able to move through the gel pretty quickly. The really big fragments are going to get lagged behind way back here. And so eventually what you have is these bands that form, all the different segments of DNA. Well, if I want to take a section of DNA and compare it, say, to another piece of DNA, are they similar? Are they different? I don't know. Let's put it in the gel and find out. And so I have known gene here, and I have unknown A. Is unknown A like the known section of DNA? Well, look, when I apply the same restriction enzymes, they don't create similar strands, so they're not similar. This is used at crime scenes. This is used, has lots of different applications for understanding the human genome and how we compare it to each other. One final thing here is on the Human Genome Project needs to be mentioned. This happened in the 90s and early 2000s, where the entire human genome was mapped. Anywhere from 25,000 to 40,000 genes were discovered, and where they are discovered. The problem with that is because of something like alternative RNA or RNA alternative splicing, many different proteins can be made, and so we really just discovered that things are much more complex than we ever could have imagined.